Today's lecture, we're going to discuss about nucleic acids. Nucleic acid is an overall name for the DNA and RNA, and you should know what these molecules are, as well as what their functions are. In this lecture, we're going to discuss about these nucleic acids in the context of the biochemical reactions, specifically focusing on the connections between the structures and the functions of those DNA and RNA, as well as their biochemical properties. DNA, which is short for the deoxyribonucleic acid, is the genetic material of all free living organisms. So it contains the information, critical information for the sustain of the life, and also it can be inherited, as you know. Whereas RNA is short for the ribonucleic acid and primarily serves in the transcription and translation of the information. So this RNA basically works as a bridge between DNA and the proteins. And some viruses have RNA genomes instead of DNA, such as an infamous virus, the coronavirus. And nucleic acids are linear polymer of nucleotides, which means that nucleotide is a building block for the nucleic acids such as DNA and RNA. And DNA is made up of a large number of deoxyribonucleotides, which is a subtype of these nucleotides. However, the RNA is made up of ribonucleotides, which is slightly different than these deoxyribonucleotides as subunits. And each nucleotide is composed of a nitrogenous base, as you can see here as an example. It shows the adenine, and a 5-carbon sugar that is attached to the nitrogenous base, which is called pentose sugar, and also a phosphate group that is attached to the pentose sugar. And the sugar molecules in ribonucleotides, which is a building block for the RNA, is ribose. And what it is basically the same as this structure. Instead of hydroxyl group, OH, in here, instead of hydrogen atom. However, the sugars in deoxyribonucleotides, which is a building block for the DNA, has the 2' deoxyribose, which is the hydrogen atom, instead of the ribonucleotides that has the hydroxyl group here. And at physiological pH, every residue carries a net negative charge because of this phosphate group, which is a negative charge, as you can see. And the nitrogenous base, which is attached to the pentose sugar in the nucleotide, has 2 generic types. And at times, I'm going to just call it as N bases or even NB. Anyway, there are 2 types, which are the purine and pyrimidine, which looks like this in here. And you must know the structures and have to remember the structures, as well as have to memorize the atom numbering conventions here, as you can see, starting from 1 to 6 for pyrimidine, as opposed to purine has the slight different numbering, starting from 1 here on the nitrogen atom here, and then goes to 6, and then 7 to 9. So here are the common features of nitrogenous bases. First, they are aromatic heterocyclic structure, just like a benzene, which has a C6H6 ring. And I'm pretty sure you're familiar with this benzene structure, but you can see it here too. And they have conjugated double bonds, which makes this 6-carbon ring structure highly stable because the electron clouds are delocalized here, meaning that there are double bonds that is formed between 2 carbon atoms, and that can be actually moved to the neighboring carbon-carbon bond too to make the resonance structure. And also it's more like the dynamic structure, as you can see here, as resonance hybrid. And each carbons on the this nitrogenous bases have sp2 hybridization, and the unhybridized p orbitals form the p bonds, as you can see here, and that gives the electron densities, especially above and below of the plane of this 6-carbon ring, and that is basically delocalized again, give this special feature as a resonance hybrid. And these p electrons are shared, like I told you, and its consequence is that the O bonds have a partial double bond characters between the carbon-carbon atoms, and that restricts the rotation, so the ring is relatively flat and planar, and it's quite rigid too. But some asymmetry can be introduced here too by substituents of at the different positions around the ring as a non-carbon atoms. So it can be added here instead of hydrogen here, or even the carbon atoms too. It can be replaced by that. And those substituents can allow the hydrogen bonding with the other molecules in the vicinity, which is particularly important in the nucleic acid structure, especially for the DNA double helix structure, where the two nitrogenous bases are interacting with each other through the hydrogen bonding. And we're going to discuss about these interactions much more in details later on this lecture. Otherwise, if there's no asymmetry, then nitrogenous bases will be just simply hydrophobic, just like benzene. Before we get to actual nitrogenous bases found in DNA and RNA, I want to make this clear that you must memorize bases structures and their numbering systems, as well as the donors and acceptors in hydrogen bonds that is involved in the interaction between the nitrogenous bases, especially forming the double-stranded DNA. And you also must be able to draw and identify bases, and also draw the base pairs too. This is not because I want to give you a hard time on the exams, but because I want you to have the foundation to understand how the molecules between these nitrogenous bases interact with each other to form the double-stranded DNA or even RNA, or the interaction between the DNA or RNA with other biomolecules such as proteins too. So it's really critical. You need to understand the structure to better understand the function of those nitrogenous bases and different types. So they have largely two different types here. Two purines, which are adenine, can be written as A, or guanine, written as G, and also three pyrimidines. One's a cytosine, which can be written as C, thymine for T, and uracil for C. And especially for thymine, it can be only found in DNA, but not in RNA, as opposed to uracil can be seen only in RNA, but not in DNA. And there are also some minor modifications on these nitrogenous bases. Here are the examples. One is 5-methylcytosine in DNA, which has an important role in gene expression that we are going to talk about much later in this course. And also RNA has many base modifications with various functional roles. The examples include transfer RNA and also mRNA modifications for the transcriptional regulation. This table shows the chemical structure of the purines and pyrimidines. And again, I want you to memorize all these structures and appreciate the differences between the bases even within the same group as purines or pyrimidines. And also this table shows the different namings based on the what other molecules or the groups are attached to them. So the first one is adenine. If the base region, which is indicated here as X, is just a hydrogen, then it will be called as a basic name as adenine, and also indicated as just one letter A, and guanine, cytosine, uracil, and thymine, as you can see here. If this X group is replaced to the ribose or the deoxyribose, then it will be called as nucleoside, and their specific names are going to be adenosine, guanosine, cytidine, uridine, and deoxythymidine. And also the one letter indicate is the basically the same as just the basic form here as A, G, C, U, and DT for the deoxy. When there's a phosphate group that is attached to the nucleoside, especially on the ribose or the deoxyribose, that'll be called as a nucleotide, as we discussed earlier. And their name is going to be adenylic acid for the adenine, and more often used as adenosine monophosphate here, or abbreviation for that is AMP. And it's the same way for the other bases such as GMP, CMP, UMP, and DTMP. Again, I want you to get familiar with all these namings as well as the structures. Nitrogenous bases have tautomers, and as you might know, tautomers are the structural or constitutional isomers of chemical compounds that readily interconvert in their structures. So for these nitrogenous bases, they have either keto form or enol form, where keto form has the ketone or the aldehyde group, as opposed to enol form has the alcohol, as you can see here. And this keto tautomer is strongly preferred over the enol tautomer for these bases such as thymine and guanine. So these forms are called as constitutional because of the majority of this structure within the cellular system. All this tautomerization results in the formal migration of a hydrogen atom or a proton, as you can see here, from here to here within the same structure. And that is accompanied by a switch of a single bond and adjacent double bond too, as you can see here, as indicated in red. And that is have an important role, especially interacting with the other molecules from this keto or enol forms, and the hydrogen bonding too. The sugar in the nucleotide is pentose or 5C 5-carbon ring. And they're in a slightly different form in DNA versus RNA. In RNA, it's ribose, or more formally, beta-D-ribose, as opposed to in DNA, they are the deoxyribose. And the only difference is that the deoxyribose have the oxygen atom missing at this particular position, where the ribose has the hydroxyl group. And the numbering convention here is that the ribose or the deoxyribose has the numbers with the prime. So it'll be called as 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime too. And again, you should memorize all the structures of these ribose and deoxyribose, as well as the numbering conventions. And keep in mind that they have primes after the numbers on the sugars, as opposed to nitrogenous bases have only numbers. Both nucleosides and nucleotides have nitrogenous base that is covalently bound to the sugar at the carbon 1 prime right here. And these bases are going to be connected by the N-glycosidic bond via N9 of the purines and N1 of the pyrimidines. And they are color-coded in the same way here as base. And on the other side, on the 5' region, if that is only the hydroxyl group or the hydrogen, then this structure will be called as nucleoside, as opposed to if there are phosphate groups, then that will be called as nucleotide. So again, don't be confused between the nucleotide and the nucleoside. Once this nucleoside has the phosphate group, then it will be called as nucleoside monophosphate, or diphosphate or triphosphate based on the number of the phosphate groups that are attached to it. And also their abbreviation will be AMP, ADP, and ATP. And this hydroxyl group on the 5' carbon, which is exocyclic because it's outside of this pentose cycle, it gets esterified by the phosphate to get the nucleotides here. And the nucleotide cursors, like we discussed, have 1, 2, or 3 phosphates there, especially at the 5' region. But in the DNA, which is the nucleic acids, the polymer structure, there's going to be only 1 phosphate, and that is attached to it. So it's going to be always the monophosphate with the other sugar that is attached to this pentose sugar. And the phosphates are usually drawn with the 2 protons dissociated, as you can see here with the electrons indicated, because that is what is happening at the pKa around the 7, which is a physiological range. Here's a quick summary. First, the nucleoside consists of nitrogenous base and sugar, and its naming is different from nucleotide as it starts with a base name and then add osin or idine, such as adenosine, guanosine, cytidine, and so on. And the nucleotide also have the nitrogenous base and sugar, just like a nucleoside, but it also contains the phosphate or phosphate groups, which is attached to the 5' of the sugar. So type of nitrogenous base, type of sugar, position and number of phosphates can be explicitly specified. The examples here is the adenosine 5' monophosphate or the cytidine 3' diphosphate and so on. And again, you must be able to correctly name and draw all these nucleosides and nucleotides. And saying nucleotide 5' monophosphate is oxymoron because the nucleotide already contains a phosphate group or multiple phosphate groups. So adding another monophosphate here wouldn't make sense. So you should write it as a nucleoside 5' monophosphate in this case. And the short end is AUGCT for nitrogenous bases, as you know. And sometimes it also can mean the nucleosides too. And also, for some nucleotides, it can be shortened as AMP, ADP, and so on. So what are the biological significances of the nucleosides and nucleotides? First, they are building blocks of DNA and RNA, so they must be there for transferring and also inheriting information for sustaining life. And cyclic nucleotides such as cyclic AMP, cyclic GMP are important regulators of the cellular metabolism that we're going to discuss about in a later of this course. And NTPs such as ATP, GTP, CTP, and TTP are carriers of chemical energy. For example, ATP is the energy molecule, and the phosphate group within this nucleotide are involved in this function, not the bases or the sugar. Again, we're going to discuss about that in more details in a later of this course. The nicotinamide adenine dinucleotide NAD and its phosphate derivative, which is NADP, and also flavin adenine dinucleotide FAD and its derivative FADH, all participate in several oxidation reduction or redox reaction in the cells. Again, all of them such are important biological functions that we are going to discuss about that in later lectures. So how do monomer units such as nucleotides form into a polymer, the nucleic acids? The nucleotides are linked by a phosphodiester bond, which links the 5' and 3' of neighboring monomer units. And pyrophosphate, also called diphosphate, which are basically two phosphate groups attached to each other, is a leading group in synthesis of nucleic acid by polymerases, meaning that NTPs are the precursors, not the NMPs. And again, NTPs are the all nucleoside triphosphates such as ATP, TTP, GTP, and CTP. And the NMPs are the nucleoside monophosphates such as AMP and so on. And the linear polymer of the nucleic acid has a polarity due to this 5' phosphate and the hydroxyl group at 3'. And this polarity is given to the sugar phosphate backbone here. And by convention, nucleic acid sequences are written in a way that it starts from 5' to the 3' direction. And also polymerases always grow from 5' to 3', not the other direction. And when you see the DNA sequence like this, it really means that the 5' and the sequence and the 3'. Sequence of nitrogenous bases define the primary structure of nucleic acids, but the backbone itself, which is phosphate sugar, and phosphate sugar here, is monotonous, so there's no information in there, but only the nitrogenous basis that is not regular and is peculiar for that particular genes or the functions. And this sequence can be written as just a single letters indicating the nitrogenous bases, such as A, T, G, C, and so on, or can be used as this simplified version or drawing the whole structure here. And you should be able to read the nitrogenous basis as well as how the nucleic acid is structured by looking at these different versions of indications. So in this simplified version, the nitrogenous base is written on the top, and the 2' on the sugar is indicated here as hydroxyl group for the ribose, or the just hydrogen atom for the deoxyribose. And also 3' region is connected to the 5' by the phosphate. There are 3 pieces of evidence leading to the famous Watson-Crick double helix structure of the nucleic acids. First one is Chargaff's rule, which says that for DNA from any sources, the total number of adenine is the same as total number of thymine. In a similar way, the total number of cytosine is the same as the total number of guanine, even though their compositions varies between species. The second piece is that ketotautomer is strongly preferred over the enotautomer for these bases in a living system, meaning that there could be some regularity of the structure built up by this ketotautomer, as opposed to if there is a mixture of these tautomers, then there are going to be some kind of random non-polar structure that is formed by these nucleotides. And the last piece is about the DNA crystals that they got for the X-ray crystallography, and the diffraction results combined with the molecular modeling show that they're having some kind of double helix structure. All those evidence led to the fact that 2 strands in double helix have opposite polarity, which is called antiparallel, and also is right-handed. And right-handed helix is one that if you grab it with your hand, then your finger should pointing upward. And nitrogenous bases are inside of the double helix, and they make a base paired with the nucleotides on the each strands. And they use the hydrogen bonding for those interactions. But the sugar phosphate backbone is outside, and since they are negatively charged by the phosphate groups, they will interact with water and will dissolve in it. Nitrogenous bases are paired due to mutual structural complementary, and also this pairing is extremely specific as T, the thymine, always interacts with the A, adenine, not with the C or G, and it's the same for the cytosine that always interacts with the guanine, but not with the adenine or the thymine. And between the A and T, there are 2 hydrogen bonds that is formed for their interaction, as opposed to G and C has 3 hydrogen bonds there. And there's a hydrogen bonding that is formed between A and U too in double-stranded RNA, which has 2 hydrogen bonds. So consequently, the G and C interaction, which has a 3 hydrogen bond, will be stronger than the interaction between T and A. And it is important to remember that base paired AT and GC have similar overall dimensions and the geometry too. So that means DNA has very regular structure. So there are not going to be any bulges or distortions or even any like loose regions or so, because they have exactly the same geometry here that keeps this double helix with the same distance all the time. And again, you must be able to draw the base pairs and identify hydrogen bondings, donor groups, and acceptor groups within these nitrogenous bases. The formation of hydrogen bondings is fundamental to the biological activity of DNA as well as RNA. And the atoms involved in hydrogen bondings are listed here. For example, guanine has the N1, N2, and O6 that is interacting with the O2, N3, and N4 of the cytosine. And it's the same for the adenine and thymine. The central dogma of biology explains how the information flows from the nucleic acids to the protein, which is a functional group or the entity for the living organisms. So the DNA contains the information, and that can be replicated and inherited to the offsprings. And from the DNA, RNA can be made through the process called the transcription. And once RNA is there, now RNA can be used as a template for the protein production, which process is called as translation. Replication of DNA has the semi-conservative properties, which means that both of all the strands are going to be used for replicating and generating new strands of the DNA. And once the new strands are generated, each of them will hybridize and base pair with the old strand, and each of this pairing will be inherited to the daughter cells, which means that only one of it is the newly generated and the other one is the old one. So that's where the semi-conservation comes from. And another property of the nucleic acid is collinearity. The definition of collinearity is the condition in which some of the independent variables and the entities are highly correlated. So you can actually see that the high correlation between the DNA, mRNAs, tRNAs, as well as proteins. Again, mRNA is the one that have the information in the type of the sequences of nucleotides, which have a specific sequence here, and through the transcription, messenger RNA is generated, which is in the same direction and contains the same information, which has a high correlation between them. The only change is from the T to U here. And the transfer RNAs, the tRNAs, comes in here for the translation into the amino acids and basically the polypeptides of the proteins here. Again, all the information is contained here too, quite specific and also highly correlated between the tRNAs and the amino acids as well as protein too. You can separate the nucleic acids such as DNA or RNA fragments according to their size by gel electrophoresis. And the size of DNA or RNA is basically the same as their length because of their linearity. So what you do for the gel electrophoresis is that you place a cassette inside of the buffer, and you also place the agarose or the acrylamide gel inside of this cassette. And you also design the gel in a way that you can load the multiple samples containing DNA or RNA fragments there. And you also place the cathode and anode at the opposite side of this gel. Now you're ready. So you're gonna load your samples, which contain the multiple DNA fragments. And once you turn on the electricity here, the electricity will go through the cathode and anode, and the anode will work as a plus end. And since DNA or RNA fragments have a sugar phosphate backbone, they are negatively charged, so they're going to migrate towards the anode, which is the plus end here. And depending on the size of those DNA or RNA fragments, they're going to show the different migration rate there. By heavy or the larger DNA fragments stay on the top, as opposed to the small DNA fragments will migrate much faster to the downward. So you can actually separate these different fragments there and see the patterns and can tell if those certain samples have the same constituents for the DNA samples or the fragments or not, as well as you can identify certain DNA fragments by breaking down and see the patterns of them by the gel electrophoresis. Molecular cloning is a process to generate a population of identical DNA molecules, and restriction enzymes are a critical player for this cloning process. A restriction enzyme cuts only double-stranded segments that contain a particular sequence, and a type 2 restriction enzyme, which is endonuclease, makes its incisions only within the sequence known as recognition site. And this recognition site has an interesting property where, when you see it here, it has the twofold symmetry axis, and around the axis it has a mirror images, as you can see here, CTA, and it also goes to CTA here too. And also, from 5' GAT can be seen here too as a GAT. And this structure is called as palindromic sequence, or the palindromic structure. And some enzymes make these strands incision immediately right here, opposite one another, producing blunt end of the DNA fragments, as opposed to many of other enzymes make slightly staggered incisions, resulting in sticky ends which is basically have one strand have some protrusion of the some hanging single stranded nucleotides here. And it can be hybridized to the other complementary sequence. That's why it's called a sticky end. Again, molecular cloning is the construction process of a recombinant DNA molecule. And recombinant DNA is basically multiple small DNA fragments that were originated from the other DNAs, but they were put together to become a one big chimeric DNA. And to do so, the phosphodiester bonds that was broken by the restriction enzyme should be reformed. And that can be done by the enzyme called ligases. But these ligases work only when the end of those fragmented DNA have the complementary sequences. So this diagram shows the simplified version of the molecular cloning, where you start with the cloning vector, which is a circular DNA, also called as a plasmid, that can be replicated once it's inserted into a certain species, such as bacteria. And you have a foreign DNA, let's say you want to put these together, especially by cutting some of the regions of the foreign DNA, then you should use restriction enzymes specific to the region that you're targeting, and it'll generate some of the sticky ends, or at times blunt end too. But you should have the same sticky ends for this cloning vector by treating with the same restriction enzymes. Once you have this ready, then you can purify this cut cloning vector as well as this fragment and put them together, and using the ligase, you can build the chimeric DNA that later on you can put it into the bacteria systems or some other systems for the replication of this particular DNA fragment. These are the common features of cloning vectors or the plasmid. The first one is origin of replication, which is unfortunately not indicated here, but the origin of replication is the place where the replication of a cloning vector initiated once you put this vector into the system where the replication is allowed, such as bacteria. And the other one is the selection marker, such as antibiotic resistance, to identify positively transformed cells. And transformation is a process to put this cloning vector into the cloning system, which is again bacteria or yeast. And transformed cells are the cells, the yeast or bacteria, that contain these cloning vectors. And also there's another feature, multiple cloning site, also called as polylinker, which have a lot of these restriction enzyme recognition sites that you can utilize for cutting and putting into some of the genes in your interest. Polymerase chain reaction, or PCR, is such an important and crucial tool that is used every day in biological researches. And also, upgraded version of this PCR is used for testing the coronavirus. So this PCR is used to amplify specific regions of DNA by putting the DNA fragments that you're interested in to amplify, and also primers, which are short artificial DNA strands, usually only about 20 base pairs long or so, and they have complementary sequence to the beginning and end of the DNA fragments to be amplified. So you're putting two primers, one's binding to the beginning, one's to the end, and once it's bind there, then now the DNA polymerase comes here, bind to the primers, and then the elongation process starts for the synthesis of that DNA fragments that you are interested in, from the one primer to the other primer. And this process, from the annealing to the synthesis, gonna go multiple times, and that's why this is called as chain reaction. Let's talk about the procedure of PCR. So the PCR process usually consists of a series of 20 to 35 cycles, and I briefly talked about this one cycle in a previous slide, and each cycle consists of these three steps. which are also written here too. The first step is the denaturing process, where the double-stranded DNA is heated to roughly 95 degrees C or so to separate these two strands. It's just the peeling off all the base pairing between two strands. And it usually takes about 1 to 2 minutes or so. And the next step is annealing. So after the separating these DNA strands, now the temperature goes down to 45 to 65 degrees C, so that the primers can bind to those single-stranded DNAs depending on their complementary sequences. And the time takes roughly 30 seconds to 2 minutes or so. And the last step for that cycle is the elongation process, where the DNA polymerases copies the DNA strands starting at the annealed primers. So both this direction and this direction too. And their temperature depends on the polymerases that you're using. So for example, you use 72 degrees C for Taq polymerases, which is one of widely used polymerase for PCR reaction. And this time also depends on the efficiency and efficacy of this polymerase, as well as the length of DNA fragments that you're amplifying. And the rule of thumb is that you take 1 minute per 1 K-base pairs. To identify individual nucleotides within the DNA fragments, the chain terminator sequencing, or also called as Sanger sequencing, is used. What it does is basically use the same kind of principle as the PCR, use a primer, but instead of using 2 primers, it uses only 1 primer, initiating from the region of the interest, and then use the mixture of just the NTPs, which are the building blocks for those DNA fragments, and also the dideoxynucleotide, which is a chain terminating nucleotide. Whenever the DNA fragments will have this dideoxynucleotide, it won't be able to make it elongated anymore because of the dideoxy region that can't be form the phosphodiester bond with the other nucleotides. So using that property, you're gonna end up having a lot of DNA fragments with the different sizes, and by marking that dideoxynucleotide, you can actually identify this last nucleotide by going through the polyacrylamide gel or a narrow glass tube, which is called a capillary, filled with the viscous polymers, to identify individual nucleotides from the last point towards to the first region, the 5' region of these DNA fragments. So here it shows the difference between PCR and the Sanger sequencing. So for the primer extension for the PCR, the DNA polymerases bring in the NTP, which has triphosphate groups there. And by having the hydroxyl group for the 3' region of the elongated DNA fragments, they can form the phosphodiester bond here and keep going to make this DNA fragment elongated. But for the chain terminator, which is a dideoxynucleotide, now it does not have this hydroxyl group on the 3' of the base of the sugar here, so it cannot form the phosphodiester bond anymore. So all the chain reaction will stop here, or the elongation will stop here. Denaturation and renaturation of DNA is a reversible process. So from the native state, you can get this DNA denatured by treating the hydroxide or heating those molecules. But by removing those factors, you can actually get those DNA renatured into the native states. You can use a spectroscopy to analyze DNA, because DNA has a property to absorb UV light with a major peak at 260 nanometer. And this absorption is useful because it varies with the structure of DNA, such as a single-stranded versus double-stranded DNA, as well as RNA too. And the extinction coefficient, which is the amount of light that is absorbed by the molecule, depends on the structure and relates directly to how much light that the structure absorbs. So this property of the DNA allows to relate the structure and absorbance to concentration of that. And of course, observance of the light is directly related to the extinction coefficient. So double-stranded DNA has a low extinction coefficient, as opposed to single-stranded DNA has a higher extinction coefficient. And you can see hyperchromic shift when you plot the relative observance of the nucleic acids at 260 nanometer as a function of temperature. And as you can see here, there's a dramatic change right in the midpoint in terms of their observance. Especially at the lower point, they have low observance, meaning that when the two strands are come together, they quench the observance. But once they pass this midpoint, which is higher temperature, so the DNA is denatured, the quenching disappears, so the observance rises 30 to 40%. And that means that around this midpoint, which is called the Tm, or the melting temperature point, there's a rapid change of the the native form to denatured form of the double strands. And that also tells the cooperativity of that double strands. So once the one of the base pairs broken, then all of them is going to be broken at the same time at these higher temperatures to get denatured dramatically. And again, this Tm, the midpoint of this hyperchromic shift, is the melting temperature point at which strands are half denatured. So when you monitor the Tm, the melting temperature of the double-stranded DNA, there are 4 factors that are affecting the melting or denaturation temperature. First one is the GC contents, also the AT contents. When the GC contents, the percentage of GC is high, it will raise the melting temperature, and the other way too. And the salt concentration also affects the melting temperature, as high salt will raise the Tm value, and also sequence length will affect it too, as well as gaps between the two strands that are held together. So if there are so many gaps, then it will be easily denatured, so the melting temperature will go down with the number of mismatches too. And as the GC has the stronger interaction between the base pairs, again, remember that there are 3 hydrogen bondings for GC, but only 2 hydrogen bondings for AT. So the AT region will be denatured first, and then the GC after that as the temperature goes up. So again, here are more details about the factors that affect the Tm values. First one was again the GC contents, where the number of hydrogen bondings different between the different base pairs, which will affect the Tm overall too. And the GC content is highly varied from 22 to 70% depending on the organisms that you're working with. And also length of the DNA will affect the Tm value too. And for the salt concentration, the lowering the salt concentration removes the ion that shows the negative charges of DNA, because of the negative charge is important for the DNA stability, as the sugar phosphate backbone is negatively charged. Thus, low salt will promote the denaturation of DNA at relatively low temperature, and the increasing salt concentration will do exactly the opposite there, which raises the melting temperature. And there are more factors that affect the Tm values too, such as the organic solvents in the DMSO, or the dimethyl sulfoxide. And also formamide disrupts the hydrogen bonding between the DNA strands. Thus, it actually lowers the Tm values there. And also pH changes the Tm values by disrupting the hydrogen bonding too.